Hello everybody and welcome to the second in the series of talks in which we'd like to consider the Genesis account in the light of modern science and asking the question, is it still reasonable to believe in the first few books of the Bible in the light of modern science? And this, as we say, is the second in the series. In this presentation we'll be looking at the natural world and particularly the complexity of life. And when we see the wonders of the world around us and our diversity of life, we have to ask the question, how did all these many life forms get here? Is it the result of time and chance as described by science, or was indeed it all created by God in 6 days, 6,000 years ago? So these are our choices. The tree of life is that a reasonable way to consider the evidence of the life around us, or is faith in some would say old-fashioned and out-of-date book still reasonable? We want to look at this choice in the light of what science tells us. And we'll also consider, is there a middle way? Is it possible or reasonable to accept both that God created the world and science explains how it got here? This is often called theistic evolution, and we'll consider that towards the end of the series. So that's a really big subject, and we'll never be able to look at all aspects of it. But we hope to open your minds to some of the possibilities so you can start to consider it for yourself. We're going to split our subject down into various titles, and there will be some overlap between the subject areas. But until we get to the end, I don't think we can realistically draw any conclusions. So we hope that you'll stay with us through the series. In the last presentation, we looked at the age of the Earth and concluded that the firm account that science gives us, it might not be quite as strongly presented as it is suggested. And we'd like to point out at the beginning that we don't profess to hold all the answers to all the problems, but we'd also respectfully suggest that neither does science. We'll present these problems as we come to them, and hopefully you'll be able to make your own mind up and come to your own conclusions as the series progresses, though clearly we hope that with us you'll come to the conclusion that the Bible account is a reasonable way of looking at the evidence of life we see around us. So, to our consideration for this presentation, the complexity of life. And we want to start our thoughts with biology here, as it puts everything else into perspective that we'll consider later, but also it builds on the considerations of the last class. And the current position that science takes is that life started about 3.7 billion years ago with simple bacteria, and more complex forms of life, as we might recognise them, came around in the Cambrian explosion about 540 million years ago, and then evolved through natural processes into all the life we see around us today, even our very selves. And the alternative position is that God created the world in 6 days, 6,000 years ago, as explained in the Genesis account. So on the face of it, these two views are not compatible. How can we take seriously a Bible account in the light of scientific discoveries? So let's look, to start with, at the scientific position in a little more detail. Now, as we talked about last time, neither of these two men were the first to propose their theories, but they have become famous and associated with them. And perhaps it's the work of these two, more than almost any other, that has led to the position of the current scientific understanding today. Charles Lyell's book on the principles of geology laid down the long periods of time that gave room for Darwin's theory of evolution as the origin of species. The premise of their work was that both men started with the idea that it was natural processes and not a creator that made the world and everything that we see around us. So Darwin proposed that given enough time and the natural gentle pressure of competition and chance mutations, that they in themselves were sufficient to life to develop from simple life forms to the complexity we see today. Now, we won't be looking too much about the development of life this evening, but we'll keep that for another time. But the tree of life is perhaps more than any other image symbolises this idea. And phylogeny is the science of putting things into an order. And you could do this in your kitchen, empty all the drawers and cabinets and put everything in order and make a tree that represents how these relationships are come together. And this is what science has done. They've looked at the various animals and their aspects in great detail and decided how they are related to each other and come up with a tree of life. And the figure on the other side is Darwin's original sketch with "I think" across the top as he pondered this problem and these ideas. So is that a reasonable explanation for the life that we see around us today? Charles Darwin, in a letter he wrote to his friend Joseph Hooker in 1871, gave these words: "But if in some warm little pond, with all sorts of ammoniatic acids and salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to go under still more complex changes." So this is the position that modern science takes, or at least one of the main ones. And it's probably the notion that you have for how science explains how life started. It's been put over so many times and so well reinforced in our minds that we almost accept it as fact. So let's look at that proposal and see if it's feasible or not. Now, back in Darwin's day, this gentleman, Gregory Mendel, who lived at a very similar time to Darwin, was doing some experiments with pea plants and is known as the father of modern genetics. Now, his experiments looked at the way aspects of the pea plants were passed down from one generation to another. And his theory, the median line inheritance, is still the basis of much of today's science. But it wasn't until the 1940s or the 1950s that we started to realise that DNA existed and played such a big part in this. And Watson and Crick demonstrated the dual helix structure of DNA that we're now so familiar with only in 1953 and got a Nobel Prize for it. And the Human Genome Project was declared complete in 2003, when one complete human genome was sequenced so we could look at the instructions in DNA. Since then, there have been many experiments and more research, and now with thousands of individual genomes that have been sequenced. But all this is 130 years after Darwin first pondered about that little muddy puddle that he imagined life started in. So Darwin really had no idea at his time of the structure or the functioning of a single cell. And it wasn't his fault. They didn't have the tools back in those days available to him. But I wonder what he would have thought if he had really have understood what a cell consisted of. So in our consideration for life, I thought we'd start by looking at a simple cell. But I realised that was far too complex and we'd be here all night and into tomorrow if we tried to do that. So we can't look at that tonight. So I thought we'd look at a single organelle. Now that's a small part within a cell, a sort of a cell within a cell. And I thought the mitochondrial would be a good place to start. We've all heard of that and probably wondered what mitochondrial DNA is, but not really known. But unfortunately, what I found out was that's far too complex for one presentation also. So, lowering my sights, I decided to look at protein. So, what is protein? Well, when we think of protein, we probably think of protein, carbohydrate and fibre, because that's how we most often come across it in our day-to-day lives, isn't it? But this evening, we'd like to look a bit deeper. We'd like to look at the structure of a protein, what it is and what it does. So let's dig into that. Well, a protein is made from amino acids. Now, just bear that in mind for a moment. Now, proteins are complex molecules that do work in our bodies. They replace tissue, digest your food. Your muscles and organs are made of protein, and your immune system, well, it virtually is protein. It certainly wouldn't function without them. In fact, just about everything that happens by protein. They're like little robots or machines running around doing things. So, we've said that protein is made up of amino acids. Now, there are about 100 amino acids that are known to science, and of those, 20 of them are used in making proteins. Now, amino acids come in left and right-hand versions, just like your hands, and they're the same amino acid, but there's just different versions. But only the left-hand ones are used for making up proteins. Right-hand ones are a bit of a nuisance in this process. So let's imagine a simple protein. A protein made up of 100 amino acids, and many of them are much larger than that. And we want to think about the likelihood of this forming on its own by chance. And the formula is 20 raised to the power of 100. That's because there's 20 amino acids and 100 positions that we're looking at for a 100 amino acid protein. And when we crunch that number, we come out with the chances of 1 in 1.3 times 10 to the 130. Now that's a strange number, isn't it? What does that mean to anything? So anybody? So let's try and look at that number to understand it in a bit more detail. How big is that number? Well, 1000 would be written like 1 and 3 noughts in our normal writing, or we could, as a mathematician, write it down as 10 to the raised to the power of 3, and the 3 represents 3 noughts. So a million is 1 and 6 noughts, or 10 to the raised power of 6. And a billion, and a trillion, and 1,000 trillion, and 1,000,000 trillion, that's 10 to the 18. Now we know that 1,000,000 trillion is a silly big number, don't we? But it's only 10 to the raised to the power of 18. And the number that we're looking at is 10 to the power of 130, or 13 with 129 noughts after it. Like that. Now, it really is a big number, isn't it? But we still really can't understand how big that number is. So let's try and put that number into some context. Now, last time we talked about it being 3.7 billion years since life started on Earth. Now, how long is 3.7 billion years in seconds? Well, it's 4.1 times 10 to the power of 12. So that's not big enough by a long way, is it? How about the total number of organisms that have ever existed on Earth? Now, this is clearly an estimation because nobody's counted them. But we're talking about everything from elephants down to amoeba over the entire time that life has existed on the Earth. And that's estimated to be around 10 to the 40. So that number isn't big enough, is it? How about the total number of atoms in the cosmos? Now, cosmos, this is the whole lot, everything. The total number of atoms. Again, it's estimated as 10 to the 80. Well, that number still isn't big enough. We haven't got there yet. Okay, so let's say all those atoms are bumping into each other all the time, and we're going to estimate that every second, somewhere in the cosmos, there are 1 trillion interactions between different atoms. And then we'll go with, since the Big Bang, that's 13.8 billion years, or 10 to the 18 seconds. Now we're going to add all those, or multiply them all together. The total number of interactions between atoms since the Big Bang is 10 to the raised to the power of 110. So that's still not big enough. In fact, our number is about 130 times 10 to the 18 bigger than the total number of molecules that could have ever existed. That's the total number of atoms that have bumped into another atom since the Big Bang. So to make up one protein, well, not quite, as we'll see. We haven't got anywhere close to it yet. And the most simple forms of life need about 75 proteins. So remember last time we thought that 13.8 billion years was a long time. Well, to make protein by chance, it's not long enough. And in fact, the universe isn't big enough. And more than that, proteins on their own don't equal life. That's quite a conclusion, isn't it? So what we've been looking at and thinking about is prebiotic chemistry. So well done, you're all prebiotic chemists now. And that just means chemistry before biology. Now realistically, the chances of coming up with a functioning protein by the operation of random chance has been estimated about 10 to the 77 to 1. And the reason for this is there are a lot of proteins that will work, and we were only looking at one. And there's some work that's going on at the moment that suggests that once a complex molecule has been created by a random chance, that it can itself encourage more complex processes to take place. But the truth of the matter is that prebiotic chemistry is in very big trouble just at the moment, because we really don't know from science how life started. Now, I'm going to do a think experiment now to consider another point. Now, think experiments can be great fun and they can be really good, but also they can be really bad. And this is certainly one that we're not going to be doing in the real world. So I'd like you to think, what would happen if we put some pond water and a frog into a blender? Now, really don't try this at home. Don't be doing it. Frogs are in big trouble, and all amphibians are just at the moment with climate change. So we don't want to be encouraging anyone to do this. But just think about what would happen. So there they are, and we turn it on. We're going to end up with frog soup. Now clearly that'll be all the ingredients for life. It would be perfect, wasn't it? This is Darwin's muddy puddle, ideally created. Now think, what's going to happen to that liquid? Have you decided? Well, I think it would go smelly quite quickly, and the wife or your daughter or everybody in the house would soon be moaning, what's this horrible stuff going bubbling in the corner? And the point of this little think experiment is that biological materials on their own are not stable. So just bear that point in mind also. Okay, so we've thought about how proteins are not made. How are they made? Well, proteins are made inside the cells of your body. The instructions or the code, the recipe for proteins are kept in your DNA. And your DNA is inside the core of every cell in your body. And DNA is a continuous string of genes. And genes are little instructions on themselves. And each gene is an instruction to do something. And many of them are for making proteins. So, each gene or discrete section of your DNA molecule starts with a start signal and finishes with a stop signal, a promoter and a terminator. And the first step in this process is we're going to have to make some RNA. This is a process called expression, and a little machine comes along called an RNA polymerase, which binds with your DNA and opens that double coiled helix that we know about, and moves along. And as it does so, it creates a new string called RNA. Now this string isn't ready to be used yet. It needs modification, and this is in a machine called an intron splicer, and that itself is made of RNA and protein. It cuts out sections of the RNA that are not required and puts on a cap and a tail, and now we have something called messenger RNA or mRNA. Now the mRNA has to leave the core of your cell through pores and enters into the ribosome. This is the area, the main part of each cell. So this messenger RNA contains all the information needed to make the protein. These are contained in groups of 3 letters called nucleotides. And each one codes for a different amino acid. There are 65 of different messages, and most of them are amino acids with a start and a stop and a few other minor signals as well. So that's our messenger RNA. Now this has to bond with another little machine. This one's called the ribosomal subunit. It assembles with our mRNA and starts to make up the amino acid. Now each amino acid is brought along to this ribosomal subunit by its own transfer RNA molecule, another little molecule that includes a message on the bottom with these three coloured feet that you can see, and it holds and brings a particular amino acid along with it. Inside the ribosomal subunit, the transfer RNA binds with a messenger RNA when the message codes match with each other. The ribosomal subunit has three sections. We'll call them in, bond and out for simplicity. As it moves along the RNA, a string of amino acids is formed. So here we have a few slides that show that process. One, messenger RNA bonded with the transfer RNA there. The next one comes in, it bonds, the machine moves along, one's ejected, another one comes in and bonds, and this continues to happen until the whole string is made up and we reach the stop signal. And then the release factor comes along to tell the machine that the job is done. And now we have what's called a polypeptide. It's not a functioning protein yet, but it's a string of amino acids. So the polypeptide has to move on, and it needs modification. Now different polypeptides go to different organelles within our cells. And we're looking here at a digestive enzyme which will go to the endoplasmic reticulum, which is a big word. And then it goes to the Golgi apparatus. And in these organelles, the polypeptide is folded and finished and made into a functioning protein. And that's how a protein is made in the cells of your body, and is going on right now whilst we watch this presentation. So what have we learned? We've learned that we need DNA to make RNA. We've learned that we need RNA to make a protein, and actually we need proteins for DNA, because otherwise, unless all of life is there, then it's just not stable, as we thought about in the frog experiment, and we said the protein helps to maintain the DNA. It's worse than that though, because proteins are made from amino acids, as we've been talking about all along, and we as animals can't make our own amino acids. In fact, plants make them for us. So is it just a happy coincidence that on the third day God created plants, and then life in the sea on the fifth and land in the sixth? I guess I always thought that God made plants first so that the animals had something to eat, but it looks like God also made them first so that we had the base materials to make our own bodies out of. So we've considered that the likelihood of a functioning protein forming by chance is about 1 in 10 to 77. Realistically, that's not a reasonable possibility. And in fact, as humans, we need about 100,000 different proteins for our bodies to function. Remember, a simple life form needs 75 proteins. So Darwin, when he started this process, imagined that life could start on its own in a puddle with the right conditions. But in the light of modern science, what we found is that actually is not a reasonable conclusion to come to at all, with the evidence that we now know that have only been found out in our lifetimes. So the current position by science is very difficult to come to the suggestion that life started by chance. In fact, science really hopes that at some point, as we learn more, that they will learn how life started. Realistically, then scientists have faith in their own process, that one day they'll understand how life started. But the real thing that we've learnt is that the more we learn about life, the less likely it is that life could have started by chance. It's been said that if Darwin came up with his idea today, it would be rejected, and there are many scientists that are not happy with the idea that evolution is a sensible solution to the starting of life. So which you believe? It depends largely on what you want to believe in, because faith is required for both. Science has given us some possibilities, but the odds are very long. The warm little puddle idea really doesn't look sensible anymore. So, have we proved that God made the world and everything in it in 6 days? Well, I don't think so far. But in the last presentation, we chipped away at the foundations, the bedstone, the scientific position that the world is a very old place. And this time we've suggested that, well, actually 13.8 billion years isn't long enough, and the universe isn't big enough for things to have happened by chance. And there are no natural processes that come close to explaining the origin of life and the complexity of even the simplest of forms of life as we currently understand things to be. In fact, not even the simple precursors to life, a protein, have been found could reasonably have started by prebiotic processes. Perhaps you might conclude that as science explores the natural world, it has led us to the position that it is more difficult to believe in the origin of life by chance than it is by the origin of life to be by a creating act. And we'll touch again on these themes as we go through these presentations, so we hope that you'll join us again. So in the first presentation we looked at the age of the earth, and now the complexity and the origins of life. Next time we'll be looking at the record in and of the rocks that we stand on literally. And then we're back to biology and chemistry for the fourth one, and then the fifth presentation hopefully we'll be drawing some conclusions of all the evidence that we've considered. So we hope that you'll join us again. And each time we'll be bringing up a list of references that I've used to pull these presentations together, and a reading list also if you'd like to take your study further. So please take a screenshot of that if you'd like. I hope you've enjoyed our presentation.